Hi, I'm Dr. Alexandra De Lima, and I would like to present my poster on antimicrobial sensitivity of Salmonella species isolated from horse feces in Malaysia. Salmonellosis is a recognized cause of enteric or systemic disease that causes anorexia, pyrexia, and diarrhea in several species, including horses. It is caused by a variety of strains of Salmonella species. There is a risk of nosocomial infections and zoonotic transmission with salmonellosis. Additionally, there is a major concern with the spread of salmonellosis with the prevalence of antimicrobial resistance due to the widespread use of antibiotics. While it would be ideal to appropriately obtain samples for culture and antibiotic susceptibility testing in horses with suspected salmonellosis for infection, this is not always practical in many cases. While waiting for laboratory results, it may alter the clinical outcome. The usual turnaround time for salmonella isolation and identification takes about 3 to 5 days. In these instances, antimicrobials would be provided empirically based on education and knowledge, previous experience, and the availability of antibiotics. The results of antimicrobial susceptibility testing are crucial for assessing the frequency and trends of antimicrobial resistance to different antimicrobials, as susceptibility profiles differ from region to region. The results of the study can provide information as to whether antimicrobials are appropriate for usage in a particular region. The objective of this study is, while there are several published reports of multi-drug resistant Salmonella species in horses throughout the world, Malaysia lacks this information. Therefore, this study intends to determine the incidence of Salmonella species antibiotic resistance in Peninsular Malaysian horses. So the study randomly sampled 82 healthy horses from 9 stables in Peninsular Malaysia. The horses were determined to be healthy by physical examination. Samples were collected where fresh fecal samples were collected as a grub sample from the rectum of the horse, and fecal sampling was repeated daily for 3 days. All samples were subject to routine bacterial culture procedures for Salmonella species, where samples were pre-enriched in buffered peptone water, enriched in Rappaport bacillidiasis broth, followed by culturing onto brilliant green agar as well as xylose lysine deoxycholate agar plates. Identified colonies were then subjected to biochemical tests for the confirmation of Salmonella positive species. So all the positive isolates were tested for antimicrobial susceptibility testing using the standard disk diffusion method as described by Bauer et al. In 1996. So a summary of this process is where 2 or 3 positive colonies were selected from the nutrient agar using a wire loop and were mixed into a test tube filled with about 2 to 3 ml of sterile saline and mixed using a vortex mixer until the density was visually equivalent to that of the McFarland standard. The bacterial broth suspension was then streaked evenly in 3 planes using a sterile, non-toxic cotton swab onto the surface of a Mueller-Hinton agar plate, and it was left to dry for 2 to 4 minutes. The antimicrobial sensitive discs were then placed on the culture using a disc diffusion dispenser. The antibiotic sensitivity discs that were used included penicillin 10 micrograms, streptomycin 300 micrograms, gentamicin 10 micrograms, and enrofloxacin 5 micrograms. The plates were then incubated for 37 degrees for 24 hours, and the size of the inhibition zone was interpreted as sensitive or resistant according to the Clinical and Laboratory Standards Institute guidelines of 2008. So the results of this study revealed a prevalence of 2.4%, where 2 horses were found to be positive for salmonellosis. These horses were from different stables. 1 horse had 2 positive fecal samples on 2 separate days, while the other had only 1 fecal sample positive for Salmonella species. The antimicrobial sensitivity testing from Salmonella isolated from horse feces demonstrated a sensitivity to streptomycin and gentamicin with resistance to penicillin and enrofloxacin. The antimicrobial sensitivity testing has been performed and reported in many live animal samples for Salmonella species, such as in chickens, ducks, other avian species, tilapia, cats, dogs, pigs, and snakes. This is the first time antimicrobial sensitivity testing has been performed in horse fecal samples positive for Salmonella species in Malaysia. In other studies with other species of animals, it has found to have sensitivities to streptomycin at 32.4 to 100% and gentamicin with 80 to 100%, similar to what was found in this study. Salmonella isolates from other species have shown sensitivities of 65.6 to 100% for enrofloxacin, while all isolates were resistant to penicillin. In this investigation, all isolates were resistant to enrofloxacin and penicillin. In conclusion, based on the study's findings, it may be inferred that antimicrobial resistant Salmonella bacteria are found in Malaysian horses. Although it will be extremely challenging to remove Salmonella and antibiotic resistance, the focus should be on limiting their spread by enforcing antimicrobial prudence and monitoring trends over time. It is also essential to comprehend the existing condition of the spread of these bacteria so that an all-encompassing control strategy may be developed for the ever-increasing antibiotic-resistant Salmonella bacteria. Thank you for taking the time out to listen. I hope you have a nice day. Bye.